Dear Sir, having been a sufferer from diabetes mellitus for over 20 years, since about 20 years of age, I have naturally taken great interest in the subject, especially in new lines and treatment. During these 20 years, science has much advanced, many discoveries been made, and many theories advanced and applied, only to fail in practice. That most in vogue at the moment, the fasting treatment, seemed to offer much promise. But when examined carefully and its results compared with the ordinary cases regulated by diabetic dietary, it will be found that the final result is about equal as far as time of death is concerned, especially in cases that have been treated for some time by ordinary methods. It may therefore be asked why a patient should be subjected to continued underfeeding and his life thus made miserable. Better admit the failure of the modified Allen treatment at once and inform all sufferers. Recently, a much more hopeful line of investigation has arisen, and that is the bacteriological. As I do a little bacteriological work myself, this interests me immensely, and I hope someday to hear that the germ causing this malady has been isolated and a suitable antigen discovered. I live in expectation of this. I am, etc., J. F. Ray.
When Dr. Ray wrote his letter to the British Medical Journal in 1922, he was unaware that only a month earlier in Canada, a young diabetic boy nearing death had just made a miraculous recovery following an injection of insulin. While Dr. Ray was on his rounds doing what he could for the diabetic patients in his practice, Richard Fox was an assistant in the Wellcome experimental department. He was just 21 when the news of the great discovery began to spread. Mr. Fox is enjoying retirement at his Kentish home, but he still remembers the events leading up to the manufacture of insulin on a large scale.
Well, following the discovery of insulin in Canada by Banting and Best, work in this country started in the spring of 1922, when the Medical Research Council asked various firms to undertake the preparation of insulin on a commercial scale. And we started work at Dartford and Wellcome Chemical Works under the direction of Dr. Sidney Smith, who was the head of the experimental department. And the very first thing that happened as far as Wellcome Chemical Works was concerned was the arrival of 2 pancreas wrapped up in a piece of muslin from London, from the London slaughterhouse, and they were brought to the experimental department, and I went up to the works canteen, to the kitchen, and borrowed a small hand mincer, and we put those 2 pancreas through this small kitchen mincer. And that was the very beginning of work at Dartford and in this country on the production of insulin. And we did actually work day and night, and work went on there with the testing of all the specimens and samples that we produced. The testing was done at the Wellcome Physiological Research Labs in Beckenham. Of course, in those days, this was not very long after the First World War, it was difficult to get plants and apparatus, and we worked under some difficulties, but gradually a process was developed, and we began to produce samples which were active and had satisfactory tests. The problems began to arise as soon as the change from making it on a laboratory scale went into manufacturing scale. Of course, insulin is very sensitive, particularly to heat, and stages in the evaporation of the extracts and concentration of the insulin solutions required the input of heat to be kept to an absolute minimum. And although this could readily be achieved in small laboratory scale, on a manufacturing scale it involved engineering and other problems. Following the war, the output of insulin was increased enormously as pancreas became more readily available. And all the time that insulin was being processed, efforts were made on the laboratory side to improve the purity and to introduce modifications where they were needed to meet the requirements of different cases of diabetes. And this work on the purification of insulin has continued right up to the present.
When Banting and Best made the first insulin, they used beef pancreas. Beef is still the most popular source, simply because it is available in large quantities and gives higher yields than glands from other sources. The younger the animal, the more insulin it has in its pancreas. But it has been estimated that about 20 animals are needed to produce enough insulin to keep a diabetic going for 1 year. At Dartford, the pancreas from 3.5 million cattle are used every year. The best quality glands are frozen immediately after slaughter and delivered in refrigerated containers, 17 tons at a time. The glands are transferred to cold storage as quickly as possible and kept there until required. Insulin manufacture is a long and complicated process and begins with the mincing of the pancreas glands. As the glands are shredded, acidified alcohol is added to dissolve the insulin out of the glands. The resulting mash is then pumped out into the main extraction plant, where the solids are separated from the solution containing the insulin. First of all, the meat fibers are taken out by accelerated sedimentation, and any remaining solid matter is removed by passing the liquid through a filter press. The clear extract which emerges still contains, in addition to insulin, blood, fat, and a number of other impurities which have been dissolved by the acid. So the acidity is altered to precipitate some of these unwanted proteins, and then readjusted to minimize the loss of insulin during subsequent evaporation of alcohol. Most of the alcohol is removed, and then condensed and recovered for further use. No small consideration, since the process requires 30,000 gallons every week. Having reduced the alcohol content to 5%, the fat now becomes insoluble. And this is extracted by adding a filter aid, filtering, and then physically removing the fat. Now comes the most important part of the process. From an original 1,000 gallons of extract, 100 gallons remain. To this is added 100 weight of common salt, which binds to the insulin molecules. When the mixer is stopped, a salt cake rises to the surface. And there, at last, is the first sign of success, a layer of crude insulin. Now begins the painstaking process of purification. The salt cake is taken off the surface as quickly as possible, usually by hand, since any other method could mean that the insulin remains too long in contact with the mother liquor and so deteriorate. From this point, it is a matter of separating the insulin from the salt and purifying it in the laboratory by repeated recrystallization. Eventually, after some 14 different purification processes, the white insulin crystals are ready for analysis. Within the last decade, it has become possible to identify minor impurities remaining in standard commercial insulin after recrystallization. These new, more rigorous procedures have shown that even preparations which have been recrystallized many times are heterogeneous and contain other insulin-related protein molecules and insulin degradation products, as well as pure insulin monomer. Column chromatography separates molecules according to their size. Standard recrystallized insulin divides into 3 major peaks. Peak A, containing heterogeneous proteins with molecular weights of over 12,000. Peak B, comprising proinsulin, intermediates, and insulin dimer with molecular weights of 9 to 12,000. And peak C, consisting of monodesamido, arginyl insulin, and ethyl ester insulin, as well as pure insulin monomer, all with a molecular weight of around 6,000. Measurement detects the presence of the molecules of larger size and relatively higher weight than the pure insulin molecule. Here, a polyacrylamide gel is being made for electrophoresis, which separates molecules according to their electrical charge. Each gel receives a sample of the insulin batch, and a current is passed through which sets up an electrical field in the gels. The positive electrode is at the bottom of the gel, and since the molecules carry negative charges, they migrate through the gel. Different molecules have different rates of migration, and so it is possible to determine which molecules are present. After 2 hours, the current is turned off, and the gels are removed from the glass into trichloroacetic acid solution, which precipitates the proteins in the gels. They're then left in a blue dye overnight. If the batch is pure, insulin will show as a single blue band. Evidence that non-insulin components are a cause of insulin antigenicity has led to unwanted fractions that produce more purified insulins, which are less antigenic than standard preparations. But as Dr. Norman Lazarus, head of the Wellcome Biochemical Research Department, points out, there is so far no firm clinical evidence that insulin antibodies are harmful. After all, many thousands of diabetics have been treated with standard insulins over long periods of time without problems.
More recently, it has been found that insulins are contaminated by various hormones, and the thought has arisen that perhaps it is the contamination of these hormones and the antibodies that they produce that are responsible for the development of diabetic complications. I am not so sure that this is true. However, I think it's important that any pharmaceutical preparation is as pure as possible. And of course, it is from this point of view that Wellcome have decided to go ahead and manufacture purified insulin. But our thoughts have gone well beyond purified insulin, and we are thinking of other ways to enable us to help the diabetic. What we would really like to do is to develop an insulin preparation that could control the diabetic blood sugar to the same levels as in normal people. The reason for this is that current beliefs state that the better the control of blood sugar, the less likely is the development of diabetic complications. Our thoughts are running on the following lines. It would be nice to develop derivatives which the diabetic person could take orally. This would mean the development of insulin preparations that are resistant to enzyme degradation in the gastrointestinal tract and that can be absorbed in such a way that the action mimics a secretory pattern of the endocrine pancreas. We would also like to get away from using animal donors for pancreas from which to extract insulin. How much nicer it would be if we could produce for our diabetic population human insulin instead of beef or porcine insulin. And I guess the only way we're going to develop sufficient quantities of human insulin for production purposes is by genetically engineering bacteria to produce it. Most of what I've been talking about is, of course, for the future. What of the present? We here in Wellcome believe it's important that we deliver to the diabetic population of this country and other countries the best insulin preparation that we are capable of manufacturing. And it's for this reason that we are now launching our purified insulin products.
But it's not just a question of greater pharmaceutical purity. The experience of diabeticians with many different purified insulins in recent years has confirmed tangible and well-documented advantages for some groups of patients in the use of such preparations. Purified insulins have been shown to reduce the incidence of pain, swelling, and other local cutaneous reactions at the site of injection. Generalized insulin allergy, a much rarer complication, is a strong indication for such preparations, though the response is less predictable. Lipoatrophy, which is the commonest long-term adverse reaction to standard insulins, disappears when patients are transferred to purified preparations, and rarely develops in those treated with them only. And lastly, there is a clear indication for highly purified insulins in high antibody-related insulin resistance, where they may help to reduce the dosage requirement. Because of their lower antigenicity, they may also control new insulin-requiring diabetics at a lower dose than would be required with standard preparations. Purified insulins are thus becoming increasingly used and recommended to resolve certain specific complications of insulin therapy encountered by some diabetics on standard preparations. In order to forestall the development of such complications, there is now a trend among clinicians to start newly diagnosed insulin-requiring patients on purified preparations, particularly children. Purified neutral insulins from Wellcome, Nusulin, Nufane, and Nulenti represent a further significant development in the company's long experience in insulin production. Experience dating back almost 60 years, and beginning with the manufacture of the very first commercial insulin only months after Banting and Best's momentous discovery. Complementing the standard preparations, they're the latest additions to the range of Wellcome insulins, each of which undergoes rigorous quality controls to ensure the highest possible standards. Just one more step in the continuing effort to help the diabetic lead a normal, active, and full life. New and better advances must come. And that's the aim of all Wellcome research.